Hello, you're listening to the Cane Matters podcast, proudly brought to you by Sugar Research Australia. Today we are coming to you from SRA's Woodford station. Nestled beside the Glasshouse Mountains of Moreton Bay, Queensland, the Woodford Pathology Research Station lies deliberately off the beaten track. Its strategic isolation isn't by chance. It forms a vital biosecurity buffer, allowing SRA researchers to safely study and manage existing high-risk diseases of sugarcane without threat to the broader industry. It's here that world-leading science works behind the scenes, bolstering the disease resistance capabilities of SRA's variety breeding program, while also responding to emerging pathogen threats. Depending on which stage of selection a clone is in as it progresses through the variety breeding program, it's screened each year for all major diseases of sugarcane: smut, leaf scald, red rot, mosaic, Fiji leaf gall and ratoon stunting disease in Woodford, and for Pachymetra root rot in Tully. And as you're about to hear from Dr. Shamsul Bhuiyan, who is the Manager of Biosecurity and Disease Screening here at Woodford, the station is leading the world in their vital work. Welcome, Shamsul.
Thank you.
So, Shamsul, Woodford plays such an important role in our breeding program at SRA. Tell us about what happens here at Woodford.
So Woodford plays an important part in our variety development program. We know that each variety can increase the productivity and the industry profit, but also we need to make sure that we respond to the new threat in a changing environment. Like this climate change, there's new threats coming. So the resistant variety in many cases are the only way to control the major disease and sugarcane. So all the variety going through our breeding program must test it for major diseases for their resistance. No variety has been released without testing for the major diseases like smut, Pachymetra, mosaic, red rot, and leaf scald diseases. So they are very important disease. Without this testing, we can't actually release any variety. So this is a bigger role, important role.
And is the testing carried out from the very first trial stages?
It depends on what type of diseases. So some diseases like Pachymetra or smut, the testing carried out at the beginning, they call it early in the selection program from the breeding program. But for some of the diseases like mosaic or red rot, it usually the advanced clone or the clone just about to release.
So that would be the final assessment trials?
Yeah, that would be the final assessment trial.
How long has Woodford existed?
This Woodford station, it started in 1997 and it started operations in 1998.
We're sitting at the station here today and looking around. Obviously, it's a very isolated station. There's good reason for that, though.
Yes, this is a very isolated station, and also actually Biosecurity Queensland given is a status of Biosecurity Zone 6 because the work we are doing here with pest and pathogens, some of them don't actually present industry, so you have to make sure there is no escape. And that's what this station is surrounded by the mountain. So we have to make sure that what we do actually remains here. We have a very strict rule, come clean and go clean. If anything comes here, you have to go clean. No plant materials allowed to actually take outside of the station.
So biosecurity obviously is number one priority because we need to protect industry from the diseases which you introduced to the plants here.
Yeah, we introduce from here, and also some of the diseases may not be present in our industry. Some of the diseases are localized. So the disease we test, there are different type of diseases. Say, for example, mosaic disease or sugarcane mosaic disease, it only present in southern Queensland. Fiji leaf gall, we never seen this disease last 10 years, so we never seen outside any commercial field.
So how does the system work with the breeding teams? How the breeding teams actually present you with their samples?
Each breeding team, they send their sample in batches. So send their test clone as a bundle of canes, and they send it to here, their clone from various stages. It depends on the disease, as I mentioned before. And we usually ask them to send it like a intact, no trashing, nothing. So we don't want to actually any damage because of the transport of their bud or any quality of the cane. So at the station, we then need to trash the cane, and then we cut them into one budded or one eye sets. And then after that, it's a different thing. It depends on the diseases. For smart, we inoculate them straight away into the smart spore suspension. For other diseases, we don't inoculate. But regardless, there's after one eye set, we put them in a trays. Then trays actually go into a germination chamber. We put them in around 31 Celsius temperature, around 98% humidity for 6-7 days for germination. And also this is the temperature good for the sugarcane smart. We can get good infection with this kind of temperature and humidity, and also good for the cane to actually grow. So after 6-7 days, we take them outside and put them in a 6-pot trays, and then we plant them in a 6-pot trays, most of the healthy cane, and then we put them in our glass house. From the glass house, they stay about 2 to 3 weeks time, depends on the space, and then we take them to outside benches. So from the outside benches, they leave it for another maybe a month to actually harden up this seedling. And then after that, in August, we started to planting them in the field. Then the whole process, it starts. So as actually one batches goes in the glass house, another batch started to do the operation like a inoculation, and another batch is this. So this like a kind of a production line happening.
So once you have these clones planted both in the glasshouse and in the field, you're introducing each of these diseases to these clones, but are you introducing every disease to every single variety, or are you only introducing them to the diseases that are known to exist in the region where the variety is going to be potentially grown?
So it depends on actually what diseases. For smart, we introduce for every one. For Pachymetra, same. And for some of the diseases like leaf skull, for every reason. For mosaic, just most of the southern one. For Fiji leaf gall, most of the southern one we use because they're not actually found in anywhere in Australia. So it depends on actually which region they came from.
And how far does the testing go? Do you test them over multiple returns, or how does the testing work?
So the testing work, also it depends on the disease. For sugarcane smart, we have to retune them because we know that smart express better on retune crop. So we have to get the full effect of the smart infection. We need to retune them, or somehow we could do the simulation of rating. So after inoculation, we plant them in the field, and then we grow for 4 or 5 months, and then we slash them. We call it retuning, and then we let them grow for another 4 or 5 months, and then we do the final rating of the disease. So it's just a retuning, but most of other diseases, we don't actually retune them. We just do the plant crop.
And when you mentioned before about inoculating all these plants with these diseases, It's not just introducing them to the soil, is it? What other means are you introducing these diseases?
So we just sort of it depends on diseases. Say for smart, we introduce them spore on the bud by dipping the one I said into the smart spore suspension. smut is the disease which actually infected through the bud, sugarcane bud. So we actually dip them into the smut. So this is, and for Fiji leaf gall, we need insect vector for infection. So that means that we need to collect the insect vector, which is a plant hopper. So we collect the plant hopper from the infected plant, and then we take the plant hopper and then inoculate them on the test clone. So put them on the test clone. The test clone actually, when it starts eating the plant leaf or the sucking into the plant leaf, it actually inject the virus into the plant. It's sort of like a malaria. You need mosquito for the malaria infection. It's sort of like it take another bacteria. Yep.
So given you're using an insect in that trial work, I would imagine you're doing that in the glass house.
In the glass house, yeah.
And how are you monitoring the actual infection process?
We let them insect to eat, feed on the plant for at least 1 to 2 weeks time. So we make sure that actually went through all the plants. We can see when you still go in the field, when you take them in the field, you can see the disease actually coming out. We are pretty sure that they're actually eating, because otherwise that we wouldn't see any disease. And it hasn't come actually easily because we had to, so SRA is there. So we actually, BSCS then, we developed this process in Australia, and then they're actually now using worldwide. Lots of the screening trial is developed by us, now actually followed by other countries like leaf gall, Fiji leaf gall, even with them like a mosaic. It's actually developed by our scientists and is now followed worldwide. So this takes years and years actually developed a method. Even in nematode, it's actually developed by me actually. I developed the nematode screening method first time.
So we are literally world leaders in this trial work.
Yes, especially in disease screening. Even with the analysis and things, we are very stringent doing the analysis and selecting the plant than any other. We can proud ourselves in this.
That's an amazing achievement. You must, though, have some challenges.
One thing we have to remember, we actually working with a biological system. So there are some drawbacks. So one of the things, we do the test in one place, which is kind of a drawback for us, because the disease may be in northern Queensland or something like this. The environment is different. We can't actually replicate the environment of the northern Queensland here. But what we actually do, even after doing our rating, we always monitor in the field as well. So we take the data from the growers' field if there is any disease, epidemic or disease infection showing, and then we always try to actually see actually what is doing. We seems to be all the tests we do actually they're fairly confident last. Yeah, say for example in smart, when first started with smart, we have about 80% of our variety was susceptible to smart. Now we have actually only less than 5% actually susceptible to smart industry. So we are pretty confident, and we can see in the field the result actually we are getting good result.
So how is your assessments on, for instance, smut translating to a reduction in smut being present in new varieties?
There is a two thing actually. One is we have some varieties very, very resistant, so you don't actually see any smut. Some variety you can get some smut, but they're very high yielding. So it's actually sort of compensate even it's in the smut. So I think that if we hadn't this testing done or the selecting, we might be hundreds of $1 million loss of productivity.
So you're weeding out the
susceptible varieties.
Yeah, at a very early stage.
Very early stage of the selection program.
How are you screening for ratoon stunting disease or RSD?
So we started ratoon stunting disease screening. This is one of the process. We used to actually use ELISA, very old method, for the techniques, sort of getting any bacterial diseases. So ratoon stunting is caused by a bacteria. So it depends on actually how much bacteria present in a plant. So the more bacteria present, that means actually more susceptible. Less bacteria is less susceptible. So you use an old method, which called ELISA method, which actually measured the antibody in the plant, but it was not reliable. So now we develop a new qPCR method. We now starting to using this one in our RSD screening method. So unfortunately, we don't have any resistant variety for RSD.
So as well as screening varieties for disease resistance, you also have two other major roles here as well. Tell us about those.
So the other major role is research on disease management and diagnostic. So in terms of research, one part of the research is management. Another part is the diagnostic. Another smart part is the improvement of current screening method. Some of the screening method, as I mentioned, is maybe 15 years old, but they are very reliable, good method, but some may be very expensive as well. So for example, for sugarcane smart, it's a very reliable method, but whether we can actually do it better. So we have some project with CSRO using molecular marker, whether instead of actually testing this one in the field, whether we can take some leaf from the sugarcane plant and do some DNA test and find out this is resistant. So it actually gives us more opportunity to screen more in a short amount of time. Also, there are some work been done on genomic. And also, we just finished a project with Griffith University using like hyperspectral image analysis. So we found that we can use the image of the plant. They take a picture of the plant. We can see actually how resistant the plant is in terms of, so we want to, they are all in the early stage program, but we want to, so this is the one part we want to, so we can do it a bit quicker, we can be it robust method, but it will cost you less. This is one part. We're trying to improve so this is our research portfolio. So we want research on better, the improved disease screening method. And the other thing we want to do actually, can you actually diagnosis using new method like sensor instead of actually putting in because most of the cases we actually look for the visual symptom. Sometimes for some diseases, it's not easy to see because disease expression usually depends on the environmental condition as well. Some diseases like leaf scald, you might get lots of infection in the leaf scald, but you might not see any symptom expression. So how do you know that it's got infection? So we wanted to try actually make like a sensor. We can actually sense the disease is there, or we can quantify the disease inside there. So we using a couple of projects like using electrochemical analysis or using LAMP testing to actually see whether actually even we don't see the symptom, but we can actually quantify the disease or we can detect the disease in the plant. So this is another thing we want to achieve. Our breeding program, screening program, we actually successfully eradicated or almost eradicated Fiji leaf gall from Australian industry. It is a major disease. So this is actually the breeding program and our screening program together. We now about to sort of declare that eradication of this particular disease from the industry, which has been present in the last 50 years or more than that. So this is actually one of the success story. Also for some other disease like leaf scald is a major disease everywhere. And also it's actually present because it can actually stay with grasses and other. But if you look at the industry, we don't have any leaf scald infection around area because our varieties are highly resistant. So we have, and even before we have like a gummy disease, sugarcane gummy disease, sugarcane downy mildew disease, which actually present in Australia. But we eradicate those diseases from Australia just using our breeding and management program. And we are proud to be part of this program. So
it's not just about to improve the tolerance of those varieties that are being delivered to industry. You're talking about eradicating these diseases as well.
Yeah. So all the diseases, they need host because they are obligate parasite. So if you have a host which is not very friendly, what do? You can't actually survive. So that's what has happened. So more resistant variety we deliver, more become hard for the pathogen to survive. So at the end, that's why we didn't see the Fiji leaf gall last 15 years. And we had like a centennial plant in every area, and we checked time to time, like highly susceptible plant, whether we didn't see actually last 15 years any disease. So that means that we have very good, hopefully we did very good job.
I think there's no question about that. Well, that was a great rundown about all the work that does happen here at Woodford, the amazing work, and like we've just said, it's world leading. Thanks for joining me today, Shamsul.
That's all right. This is my pleasure. Thank you for coming. Yeah.
And again, thanks to you, our listeners. If you'd like more information on SRA, go to our website at sugarresearch.com.au.